Hey guys, what's up? So, this is gonna be a really short video. I'm just gonna cover monohybrid inheritance and then I'm gonna be whoosh, I'm gonna be gone. Anyways, now let's this is really easy by the way. So, monohybrid inheritance is literally just a study of one singular gene, and we're gonna we're gonna do examples, okay? I've got a key here. The key says the allele big B, it's gonna the dominant allele big B, it's gonna cause an organism to be tall. The small b allele, the recessive allele, is gonna cause an organism to be um, short, right? Now let's say I got a tall plant. Let me just let's say I got a tall uh, uh, fern. What do you call this? I don't know. Anyways, let's say I got a tall one and a short one. The tall one is homozygous dominant, while the short one. Okay, I'm struggling so much to draw friends. Okay, this looks bad. Now, let's say the homozygous dominant is crossed with homozygous recessive. Now, we're gonna try to like predict the genotype and the phenotype of the offsprings. Are the offspring gonna be short? Are they gonna be tall? Um, yeah, we're just gonna try to figure that out. So for that, first we're gonna figure out the gametes. So the big B only has big B. Duh. So it can only produce gametes with the big B. We got small b, it can only give you small b, so it can only produce gametes with small b. Again, duh. Anyways, let's draw a Punnett square. So let's draw a Punnett square. Duh. Yeah, I'm tweaking. I'm sorry, I'm tweaking. Let's draw a Punnett square. Okay, not anymore. Let's draw a Punnett square. Big B, small b. And let's see. We're gonna get our offspring genotype here. There's gonna be big B, small b, big B, small b, big B, small b, big B, small b. All of the offsprings are gonna be 100% of them are gonna be heterozygous and tall. So all the offspring, they're gonna be tall. Okay, wait. Copy, paste. Yeah, I got four, one, two, three, four. Nice. All of them are big B, small b, big B, small b, big B, small b, big B, small b. Right. So this is what I'm gonna call our F1 generation. F1. So this is our F1 generation. And okay, let me just take it out. Let me just make some space. Wait. So let's let's now interbreed our F1 generation. Let's say I'm gonna breed the big B small b and big B small b, right? To produce the second generation, which is F2. Wow. let's just do it. So I got big B, big B, small b into big B, small b. It's gonna produce gametes that are big B, small b. Okay, okay, what the hell? Small b, and yeah, it's gonna, the other one is gonna do the same, right? Let's now just make a Punnett square again. So let's, we got big B. Okay, big B, small b. Sorry, big B, small b, big B, small b, and now we're gonna get the genotype. Big, big, big B, big B. I'm tired of saying big B's and small B's. Anyways, big B, small b, big B, small b, and then small b, small b. See, so one of the plan is gonna be short. So this is, okay, let me wait. I'm gonna explain. Wait, why am I dragging this? Copy, paste. So this is my F2 gen. Okay, let me get the short one. Yeah. So I got where's the blue ink? Yeah, right there. So I got three tall plants and one short. So the ratio of the phenotype is gonna be 3:1. This is F2. Okay. So when you cross uh when you cross, let's say, a homozygous dominant with another organism, you're always gonna get the dominant phenotype. And no matter the other parent's genotype, you're always gonna end up with the dominant phenotypical traits. But if you cross two heterozygous individuals, you're gonna get a 3:1 ratio where three offsprings amongst the four would show the dominant phenotypical trait, and one offspring might show your the recessive trait. So yeah, this is the infamous what do you call it? The 3:1 ratio. Now, lastly, we're gonna do something called a test cross. Now, why do we do a test cross? Test cross. We're gonna use the concepts we just learned. All right. Um, so test cross is used to done to check if the parent having the dominant trait is homozygous or heterozygous. Now, let's say I got a fern. Uh, I got a plant that is tall. Yeah, it's tall. So I know for sure that it has a dominant allele. That I can say for sure. But what I cannot say is that if it's homozygous or heterozygous, right? Because the dominant allele gets expressed either way, right? So it could be it could be your BB, or it could be B B. I mean, okay, all of them are B's. What am I saying? So this could be it could it, I'm tweaking. I'm tweaking. Sorry. So it could be big B, small b, or it could be big B, big B. We don't know for sure. But we need to find out. So to find out the genotype, what we do is we cross it. We cross it with homozygous recessive. Let's say, wait, let's say it's, let me get the correct color. Let's say if it's big B, big B, and I cross it with, for test cross, we always cross it, always cross with homozygous recessive. I cross it with homozygous recessive. Now, all of my offsprings, they're gonna show the dominant trait. Okay, all of them are gonna be tall. Let me do a Punnett square again. Let me show you how. So I got B B B big B, whatever. So I got big B small b, big B small b, big B small b, big B small b. All of them are what? All of them are tall. 100% of them will show the dominant trait. But so if 100% of the offspring show the dominant trait, you can conclude that the parent is homozygous. I wrote homoi. Is homozygous for the dominant allele. Now, what happens if the parent is not homozygous? Let's say it's heterozygous, okay? And I cross it with, I carry out a test cross, and I'm gonna make a Punnett square as well. So I got big B, small b, small b, small b, um, I'm gonna get big B, small b, small b, small b, big B, small b, small b, small b. Look what happened here. So 50% of the offspring would show dominant trait, and 50%. Okay, wait, one sec. Just, I need a little bit of space. So 50% of the offspring, half of the offspring would show dominant phenotype. So here 100% of the offspring would have been tall. Here 50% would be short, 50% would be tall, right? So we have 50% dominant and 50% recessive. So if you see a 1:1 ratio, you can confidently say that the parent is heterozygous. See, it wasn't too bad. Now, I know monohybrid inheritance is easy. And the real challenge comes when we're doing, the real challenge is going to come when we're doing the dihybrid inheritance. But in my opinion, it's not too bad either. So yeah, in the next video, I'm gonna cover dihybrid inheritance. So thank you for watching, guys. See you all in the next video. Bye bye.